Hello and welcome to Rajya Sabha Television. You're watching COVID-19 India Fights Back. I'm Frank Rausen Pereira. VLP stands for virus-like particles. Virus-like particles are molecules that mimic viruses but are not infectious. They are a very effective way of creating vaccines against diseases such as HPV, hepatitis B, malaria and more. Virus-like particles have been used as multivalent vaccine candidates against chikungunya, Japanese encephalitis, yellow fever, and Zika virus. VLPs are well known for conferring durable immune responses. Some of the most effective long-lasting vaccines are for HPV and HPV, which are VLPs. VLPs enable high-density multivalent display of antigens in a manner that closely resembles viruses with an important difference. VLPs contain no genetic material, so they are non-infectious and can provide a safer alternative to inactivated vaccines. Naturally occurring VLPs have delivered successful vaccines including Gardasil and Ceravix and also Engerix B and Recombivax HB, these against hepatitis B. However, VLPs have been difficult to use for the display of complex heterologous antigens like in the case of RSV. In this edition of COVID-19 India Fights Back, we will analyze virus-like particle vaccines. Joining me on the program today are Dr. Gagandeep Kang, Professor of Microbiology, CMC, Vellore; Dr. Madhu Gupta, Department of Community Medicine, PGIMER, Chandigarh; and Dr. Mandeepa Banerjee, Associate Professor, Kusuma School of Biological Sciences, IIT Delhi. Thank you to all my guests for joining me on this edition of COVID-19 India Fights Back. So, Professor Kang, let me start the program with you first. Let's first try and understand what VLPs are. Are there any VLP vaccines being developed as far as COVID-19 is concerned?
So VLPs are essentially the protein shell of the virus without, as you said, the nucleic material inside them, the genetic material inside them. VLPs can be quite hard to make, but if they are made, they look just like the virus, so the immune system reacts to them in the same way. For SARS-CoV-2, there are at least 19 programs that are looking at using VLPs as vaccine candidates. Of these, 2 are in clinical trials and 2 are close to clinical trials. Of the ones that are in clinical trials is one vaccine that might actually wind up in India because it is a partnership between SPI Biotech, which is in Oxford, and the Serum Institute of India. This vaccine is being evaluated currently in Australia.
Right, okay. So, Mandeepa Banerjee, let me bring you into the picture now. So, as far as VLPs are concerned, I mean, what are they really? If you could just explain to us, is it something that is naturally available? What is the process that is undertaken to add these into vaccines? How is it used really? What's the process?
So, as Professor Kang said, virus-like particles essentially mimic the surface features of the virus without actually being infectious. So most viral proteins have the ability to self-assemble. They assemble together to form a particle. That is the natural property of a virus. And in many cases, as you mentioned, for the human papillomavirus vaccines, the VLPs, generation of the VLPs are based upon this principle. A protein is expressed in a heterologous expression system, like in yeast or insect cell, or the other options are mammalian cells or plant cells or bacteria. And these proteins then come together to form a particle which closely resembles the outer surface of the virus without being infectious. So they would not be able to propagate an infection because they do not contain the genomic material. Now, these are the simplest types of virus-like particles where a single protein will self-assemble to form particles. In cases like SARS coronavirus 2, there are multiple proteins plus a lipid envelope to consider when one is designing a virus-like particle, which is native-like. So there are many permutations and combinations that one has to do. One has to consider different types of expression systems. And there could also be chimeric virus-like particles, where you're using one type of protein to form a virus shell and an antigenic protein from, for example, in this case, SARS coronavirus 2, is displayed on the surface. So there are many types of permutations and combinations that are possible. Many different types of expression systems are used, which include yeast, insect cell, mammalian cells in some cases, bacteria, plant systems. The goal eventually is to generate a particle which will display the major antigenic determinant of the pathogen that one is working on. And it is usually very safe because there is no propagation of infection.
Right. So it's much safer than anything else is what the panelists are suggesting. Madhu Gupta, let me bring you into the picture now. Let's talk about some popular VLP vaccines. What has our experience been? And do you believe that this is the right way forward as far as tackling something like the SARS-CoV-2 is concerned?
Yeah, thank you so much. So we have the experience of, as you have rightly mentioned in the beginning, human papillomavirus, then yellow fever, chikungunya, Zika virus. So there are good examples, and this shows that ultimately when we are giving vaccine, we are interested whether that vaccine is able to produce antibodies or cell-mediated immunity, which may actually will protect from the future if there is an infection, future disease. So this is what. So now, currently, what experience we have, it looks very promising. And as Dr. Gagandeep Kang has rightly mentioned, there are two such vaccines which are being in development, and they are in phase 1 and phase 2 trials. And results are quite promising, but we need to see what are the results of phase 3 trials related to these types of vaccines. So there are other options also, like viral vector vaccines. Currently, these are a forerunner, and there's this Oxford vaccine and Russian vaccine, Sputnik V vaccine, and the Wuhan vaccine, which is based upon this viral vector vaccine, wherein the gene of like you can say coronavirus is inserted into a adenovirus. So, which is a harmless virus. It's a chimpanzee virus. So, this type of vaccines are being developed to produce antibody response and cell-mediated immunity. So, this is a second type. Then there are third types wherein the whole virus is being used, wherein it could be live attenuated, where through subcultures, although the virus remains live, but it is no longer remains virulent. So it does not cause the disease, but it produces immunogenicity. So and also inactivated virus. So these types of vaccines are also being produced. And the fourth type is which is the DNA and RNA based vaccines. So these are also are under development. And we are quite hopeful that the VLP vaccine will also produce good results.
Okay. All right. So let me take the points that you're making forward and broaden the scope of the discussion a little bit. So Professor Kang, let's build on the points that Dr. Madhu Gupta has made. As far as these vaccines are concerned, there are so many different types of vaccines that various companies are trying to develop to try and get a cure, really, as far as COVID-19 is concerned. With the little limited knowledge that we have over the last 1 year, is there a clear front runner as far as a vaccine is concerned?
I don't think that you have a front runner till you have the results, unfortunately. We do have a dozen vaccines that are in phase 3 trials right now, and we expect that we will be able to get clinical efficacy results from 2 or 3 of these vaccines by the end of the year. Among those is the AstraZeneca vaccine to which Madhu referred, which is being made in India but not evaluated for efficacy in India. That vaccine is in trials in the UK, in the US, in South Africa, and in Brazil. So it's anticipated that they will have sufficient numbers for a clinical readout by the end of the year. Pfizer BioNTech also has studies ongoing where they initially started with recruiting 30,000 participants and then decided to expand to 44,000 and then are thinking about 60,000 participants for their trials. They had hoped to have results by the end of October. That didn't quite work out because you need a certain number of cases to be able to do an interim analysis of the trial to decide whether it's working well or whether to stop the trial. But it's likely that Pfizer will have results sometime at third week of November or a little later than that. Moderna has completed recruitment of its 30,000 participants, and that may also potentially have results by the end of the year. So if we talk about front runners, I would think that these would be the three. There's one vaccine that is based on a viral vector. The other two are based on RNA. There are also a lot of Chinese vaccines that have been in trials in Africa, in South America, and in the United Arab Emirates. These include inactivated vaccines and vectored vaccines, but we don't have as much information about these vaccines because the protocols for most of them are not available and we don't get regular updates like we do for the other vaccines.
Since we are here, since you've spoken about the Chinese vaccines and spoken about the UAE in the same breath, I was reading a couple of days, earlier, where the Sheikh of Dubai, in fact, took the Chinese vaccinations. So, I mean, is there anything to it? Can we say that this is probably safe, and that's why the king of the UAE is in fact taking it? I mean, I know you did mention that these are still early days and there's very little information. But what can we infer from this?
So I think the Chinese and the Russians have between them given limited licensure or certification to 6 vaccines that are being used in a restricted set of populations, and the UAE has accepted that certification and is using it more widely for essential workers, and apparently it's now being offered to medical workers as well. Now, when you look at vaccine safety, you usually look at safety at every stage of testing. So in the phase 1 trials, you'll be looking at safety in tens of people. In phase 2, you'll look at hundreds. In phase 3, you'll look at thousands of people being enrolled into the trials. All of these vaccines that have limited licensure are now at the phase 3 stage. They have been tested in thousands, in some cases tens of thousands of participants. So what this tells you is that if there are side effects to these vaccines, they will tend to be relatively rare. If you have a vaccine trial that is done in tens of thousands of participants, then it will not be able to detect a side effect that happens 1 in 10,000 vaccinated people. But certainly if there's something that happens in 1 in 100 or 1 in 1,000 people who receive the vaccine, you will be able to pick up that safety signal. The fact that we haven't seen it so far means that the side effects are more rare than 1 in 1,000.
Okay. So that's the silver lining, and that's the positive really as far as vaccine development is concerned. And since you're talking about vaccine development, Mandeepa Banerjee, what are the challenges really when we are developing a new vaccine, and especially when you're running against time, because world leaders around the world too, it's become highly politicized as well because everyone seems to be wanting this vaccine in a hurry.
Right, absolutely, because we are dealing with a pandemic, and as soon as we get an efficacious vaccine, it's better for the world. So that is why I think the politicization of this issue. But the challenges are many. So you have to generate a vaccine candidate which should have a reasonable cost. It should be very efficacious. It should be safe, and it should not have any side effects. And in countries like ours, we would have to be able to transport it to all parts of the country. We may not have adequate -80 or storage facilities. So all of that has to be taken into account. So the vaccine candidate ideally has to be very stable, so that it can be transported. And it has to be efficacious. It has to generate a good protective immune response that stays for a long period of time. And cost and other things also have to be taken into account. So it is a fair bit of a challenge. And I would say that it's not like we should have one perfect vaccine candidate. I mean, I would expect at the end of all this effort that's being put in, there would be multiple good candidates, and that is probably necessary in order to have mass immunization around the world. You need more than one good, efficacious, safe, cheap vaccine candidate.
Right. So, at some point in time, Madhu Gupta, do you believe that age and ethnicity too plays a role and will play a role as far as vaccine candidates are concerned, and what kind of vaccine can be administered to what kind of age group and what type of ethnicity?
So, thank you. So, like in case of corona infection, we have seen that those who are more than 60 years of age, they are more susceptible, and those having comorbidities are more susceptible. So, in this population, and they might also be immunocompromised. So, in this population, considering giving live attenuated vaccine could be a little this thing. Yeah, you will be apprehensive. So you would like to have a vaccine which in itself does not, which cause less harm and more benefit. So I would say like this virus VLPs and vector vaccine or mRNA and DNA based vaccines, they look better in this type of population. And regarding ethnicity, I don't think that there should be any issue, and they should be able to produce antibody response. So what we are also because I have experience of developing this vaccine, COVID-19 vaccine. So our priority group is population more than 60 years of age. So we are there is a target that we would like to recruit volunteers from this age group, so that we have enough evidence on immunogenicity in this age group as well.
Right. So, Professor Kang, since we are here then, since we are talking about different age groups and different people with different ethnicities, what should our strategy be going forward? And also, realistically speaking, by when can we expect the first truly effective and safe vaccination or vaccine?
I think the strategy for vaccination should be one that is focused on risk groups. Who are the people who are most at risk of acquiring infection and of having severe disease? So the WHO has come up with a prioritization framework for the allocation of vaccines, and there are similar efforts that are ongoing in India to align with the framework that has been suggested by WHO. And this is to give vaccines first to healthcare workers because they have the highest risk of getting infected, and to frontline workers as well, but at a later stage. And then to focus really on the elderly. There are challenges with that because you don't know that every vaccine is going to work in the elderly. But the immunogenicity data, as Madhu pointed out, should give us clues about which vaccines will work better in the elderly than others. So we'll have to think about delivery strategies, obviously, to get the vaccines to the people who need them the most. The question about realistically when will we have a vaccine is a hugely challenging one. I can tell you any date or time, and I can take a bet that it most likely will be wrong. But if we are to think about a range, then given the number of candidates that are in phase 3 trials at the moment, and as Manideepa said, it's likely that we will have more than one that is successful, we should have readouts on vaccines by the middle of next year that will result in our ability to get the vaccines to regulators and licenses from those regulators and policy in countries about how to use those vaccines. So a readout, which may happen as early as December, does not mean that we have the vaccine the next day. There are processes of licensure and processes of policy, processes of preparation for delivery that needs to be undertaken before vaccines realistically get to people. So my range is sometime in the middle of next year for vaccines that are part of the first wave of vaccines. They will not be perfect, but they will work.
Okay, right. Okay, so that's the closest as far as us getting a vaccine is concerned. Mid of next year is what Professor Kang is suggesting. All right, so Manideepa Banerjee, what's the best way forward?
The best way forward, in my opinion, is to ensure that we get multiple safe, effective vaccines that we can use eventually for mass vaccination of the world's population. Obviously, we have to start with people who are at risk, like healthcare workers, and then move on to gradually onto the other parts of the population. Till that happens, though, till middle of next year or end of next year, till we get multiple such vaccines, we have to follow the safety protocols and rules that we are doing right now, which is wear masks, maintain social distancing. I think these rules are here to stay for several months, if not a year from now. But obviously, development and safety studies, efficacy studies of multiple candidates at this point is very important, and that is what is going on.
Absolutely. All right. And Madhu Gupta, close the show for us with your concluding remarks.
So I would like to conclude that, we are quite hopeful that we will get a safe and hopefully an efficacious vaccine by, as Dr. Gagandeep Kang has predicted, by probably mid next year. And by till that time, please follow what Manideepa is suggesting. Please be safe, wear mask every time you go out of the house. Do not go out of the house if you do not have anything to do. And please maintain social distancing and hand hygiene.
Okay.
So with that remarks, thank you.
All right. On that note, then, I'll call it a wrap on this edition of COVID-19 India Fights Back. Thank you to all my guests for joining me on the program and putting things into perspective for us. And before we leave, I'd like to make an appeal to all our viewers. Remember, wear a face mask at all times when you're stepping outside the house. Ensure that you wash your hands and face regularly, and remember to maintain physical distancing when you're outside at least 6 feet. So these small measures can go a long way in combating the virus. That's it from me. See you again next time.